Hello and welcome to this video podcast for evolution and diversity. We're going to continue our discussion of bacteria and archaea, but today we're going to focus on why bacteria and archaea are important. So let's go ahead and get started on why bacteria are important. So why are bacteria important to study? Let's first state that bacteria and archaea were the first organisms on Earth. The fossil record suggests that they have been on Earth for 3.7 billion years. In fact, I like to think that bacteria and archaea owned the Earth for 1.9 billion years. For 1.9 billion years, bacteria were the only show on Earth. And ultimately, as we will learn, they are what gave rise to all the other plants and animals and fungi and protozoa that we see on Earth today. So we study bacteria because they help us understand life. They are an incredibly diverse species. There are well over 100,000 species of bacteria and archaea. Of these 100,000, we've only identified 10,000 of these species. So even though they are the smallest form of life on Earth, they are clearly the most diverse. And as we'll talk about in a moment, they are also the most numerous of all species on Earth. So let's talk about that now. Bacteria and archaea are the most abundant organisms on Earth. The total number of bacteria and archaea on Earth is 5 times 10 to the 30th. This is a huge number. To put it in another way, this represents 5 million trillion bacteria and archaea on Earth. So how much do all these bacteria and archaea weigh? Well, they weigh approximately 70 gigatons of carbon. And in case you're curious what a gigaton is, 1 gigaton of carbon is 1 billion tons. That's a lot of bacteria. To give you a frame of reference here, humans on Earth, if you added up all the humans, we would be approximately 0.06 gigatons of carbon. There's so much more bacteria on Earth than humans. And they've been around for so much longer than humans. When we're thinking about abundance, let's look at a couple other tidbits of information. There are approximately 700 different species of our bacteria and archaea in our mouths. So it's important to remember that the vast majority of bacteria and archaea on Earth do not cause us harm at all. In fact, they are beneficial to us. Almost every insect has anywhere from 100 to thousands of bacteria and archaea species inside them. Most of them are inside the guts of the insect. For instance, the ability of termites to eat wood is completely due to the bacteria that are inside of its gut that can digest the cellulose found in wood. Let's mention a few other things about the abundancy of bacteria on Earth. You could take a random tablespoon of soil and it would have billions of microbes. And when I say microbes, this includes bacteria, archaea, and other microorganisms like fungi, algae, and protozoa. Essentially anything that is microscopic. But when I say there are billions of microbes in 1 tablespoon of soil, most of that billion will be bacteria and archaea. And this is a really important point here I want to make, and that is that the most abundant cell in humans are bacteria and archaea. Within your body, on our skin, in our digestive tract, in our ears, in our mouth, in our nose, in our eyes, we have approximately 100 trillion bacteria and archaea. There isn't a cell in the human body that even comes close to that number. Without bacteria and archaea, humans simply wouldn't be human. And as we'll talk about later, these bacteria and archaea that are inside of us and are part of us, help keep us in good health. We sometimes focus on the bad health that bacteria cause, but these 100 trillion bacteria and archaea in our bodies help us stay healthy. The last one I want to mention is that bacteria and archaea live deep under our ocean floor. There are so many bacteria underneath the ocean floor that it is estimated to be approximately 10% of the world's total mass of living organisms. So here I've talked about their abundance, that when we find them, we find a lot of them. Now what I want to talk about is their habitat diversity. It's safe to say that bacteria and archaea have the most diverse habitats. They live everywhere. I'm going to list a few, but if I were going to list every place that bacteria lived, we would be here until you graduated probably. They live in soil, rock, they live in the ocean, as well as lakes, ponds, and really any body of water, even your swimming pool if you have one. We find them in the Arctic snow, the snow in your front yard. There are bacteria in it. We find them living in living organisms. We know a lot about what bacteria do to help plants and animals survive. They're found in hot sulfur springs, as well as deep sea thermal vents. It's very, very hot, but bacteria live there. It's crazy, right? We also find bacteria living in radioactive material. We find bacteria in clouds. So not surprising, we also see bacteria in rain. We find them all around our house. Every surface on your house probably has bacteria in it, unless you go around sterilizing every surface of your house all the time. When you take a shower, there's bacteria in that water. Now, it's probably not necessarily in the water itself, but your shower head is full of bacteria. So I'm not trying to gross you out. I'm just trying to tell you that when you take a shower or wash your hands in the sink, there's a lot of bacteria that are coming out. But they're not harmful bacteria. They're safe bacteria. And in many cases, they're beneficial bacteria. The next thing I want to talk about are bacteria and archaea that we collectively call extremophiles. So what are extremophiles? Well, these are bacteria and archaea that live in extreme environments. These would include the bacteria that we've talked about before that live in thermal vents. And this would be in that range of approximately 121 degrees Celsius. The actual thermal vents are hotter, but the regions where the bacteria live are a little cooler at a very cool temperature of 121 degrees Celsius, which is roughly 250 degrees Fahrenheit. So obviously still very hot. They can also live at temperatures in the Antarctic ice of 0 degrees Celsius. They can live in environments that are 5 to 10 times saltier than the ocean. They can live in very acidic places, so when the pH is less than 1.0. So on the pH scale, anything less than 7 would be considered an acid. So a pH of less than 1.0 is going to be very acidic. So any extreme environment, you're going to find bacteria and archaea. And so those bacteria would be called extremophiles because they live in extreme environments and they like them or love them. Philes means like or love. So why do we want to study extremophiles? Well, if we think about that first cell, probably was an extremophile. Because remember, those first cells probably formed in the deep sea vents or near the deep sea vents where it was very, very hot. So the very first cells were very likely extremophiles. So by studying extremophiles, it helps us understand that very first primitive life. We also are interested in studying extremophiles because we're interested in extraterrestrial life. We use extremophiles as models of potential life on other planets. If a bacterial cell or an archaeal cell can live in an extreme environment on Earth, that might tell us a little bit about possible life forms that could live on other planets that exist in these extreme environments. We also use these extremophiles in a commercial application. If you need an enzyme that works at a very high temperature or at a very low pH, then you're going to look in an extremophile to find that enzyme. So commercial applications helps us identify enzymes with unusual characteristics. A good example of this is the application of PCR, which you'll learn about when you take genetics. But PCR from a commercial application works because we use one of these extreme enzymes. Next, let's talk about the medical importance of understanding and studying bacteria and archaea. Okay, so let's go ahead and start talking about the medical importance of bacteria and archaea. The last several minutes we've been focusing on the overall abundance of bacteria throughout the world, as well as their ability to live in very diverse habitats. But for the most part, the bacteria we've talked about have been healthy. In fact, the vast majority of bacteria that we encounter are healthy and aren't going to harm us. However, there are some bacteria that will cause disease. And so we need to talk a little bit about that right now. Now, you don't need to memorize this slide. I'm just using it as an example to show you some of the diversity that we see within the types of bacteria that cause disease. Some disease-causing bacteria cause gonorrhea. Others cause staph infections. Some can cause pneumonia. Some can cause strep throat. Some are linked to tuberculosis. And some, like E. coli, can exist in both a disease-causing form and a healthy-causing form. Most of the E. coli in our body is very healthy, and we need it. But there are some types of E. coli that can cause disease and can be rather deadly, in fact. So let's begin by defining the word pathogen. And it has a pretty straightforward definition. Pathogens are agents that cause disease. These agents that cause disease, they could be one of several types. They could be bacteria, viruses, some fungi, protozoa, worms, and the last one are prions. We're not going to have time to talk about all of these right now, but throughout the semester we will touch on some of these, and as you go through the curriculum in cell biology and genetics and ecology, you'll come across some of these other examples. The only one I want to just talk about real quick here are the prions. These aren't living organisms like bacteria, fungi, protozoa, or worms, and they're not viruses either. These are misfolded proteins that are in our body. A certain protein called a prion is supposed to fold in a certain way. But if it misfolds, it can become a disease-causing agent. As an example of something you may have heard of, mad cow disease is caused by a prion. And there are some human diseases as well. But I want to focus today on bacteria. And you may have noticed that this is one of the few times that I've mentioned bacteria that I didn't also mention archaea. And the reason for that is that there isn't a clear link between a disease and archaea. So it could be that pathogenic archaea are present but not identified. As some suggestion to this idea that they might be linked to disease and we just don't know yet, we do see some correlations. We do see that some diseases are associated with a higher concentration of certain archaea species. But we haven't been able to prove that those archaea species are the cause of the disease. At this point, it's just a correlation. So we're going to focus on bacteria here, but I want to come back to this idea of a pathogen and the idea that some bacteria have been shown to be a pathogen while no archaea have. How do we go about determining if a specific bacterial species or virus, fungi, protozoa, worm, or prion is linked to a disease? Well, let's talk about that next. So to determine if a particular agent is linked to a disease, we follow a protocol known as Cook's postulates. And this was established by Robert Koch in the late 1800s. Prior to this, scientists had discovered certain agents that would later be known as bacteria. And scientists knew that these agents could do things like spoil milk or spoil food. But Koch was the first one to hypothesize that these agents, these bacterial cells, could cause disease. And disease-causing bacteria could be passed from person to person. So he established these postulates, and there are 4 of them. So to explain these postulates from Koch, I'm going to list the postulates on the left side here. And then over here, I'm going to how these postulates are tested. So the first is that the suspected pathogen must be found in all cases of the disease and not in healthy specimens. So if you were studying mice and you had a mouse like so, and you're going to be super jealous of my crazy abilities to draw mice, don't be jealous. It took lots of skill and practice to do that. And you were looking at these mice and you gave them a particular agent because you thought it was a pathogen, and the mouse died. And from this dead mouse, we find that pathogen. So maybe it's something found in the blood, and we take some of the blood out of this poor mouse here, and we look at the blood under the microscope, and we of course see, red blood cells, maybe some other kinds of cells that are naturally found in the mouse. But we also see the pathogen of interest. Maybe it's a particular rod-shaped bacteria. So this is important in showing that we can find the pathogen in the mouse that was killed, but we also need to look at a live mouse. And this live mouse, we take the blood from it, now not all of the blood, just enough of it to do the test. Taking all of it would just be cruel. And we look at this mouse's blood under the microscope, and we don't see any pathogens. We just see red blood cells and other naturally occurring cells. So this again is a good sign that what we're seeing is pathogen, presumed pathogen in the dead mouse, the sick mouse, and no pathogen in the healthy mice. Healthy in the sense that they don't have the disease, but for whatever reason, the mice I draw lack legs. I'm going to assume they're just hiding them. The second thing that must be done is that the pathogen, the suspected pathogen, has to grow in culture. So how do we do that? Well, we take this sample from the dead mouse and we plate it on a petri dish. And then we take the samples from the healthy mouse and we plate that on a separate petri dish. And we allow them to grow overnight, or sometimes a little longer than overnight. If we see on the petri dish colonies forming that are made up of the presumed pathogen, then we can have more confidence that the disease is being caused by this particular pathogen or presumed pathogen. However, it's important that we show in the healthy mice that we don't see any growth. So if this agent is a pathogen, we will be able to identify it in dead mice and not in live mice, healthy mice. And then we will be able to culture it on a petri dish if it is a pathogen, but not from a healthy mouse, only from the sick mouse. Okay, the third postulate asks that we take the cultured pathogen, or at least the thing we think is a pathogen, and put it in a healthy mouse and see if it causes disease. Running out of room here, but essentially what we're going to do is we're going to take, and I'm going to draw it up here, we're going to take a healthy mouse and we're going to take the bacteria, the presumed pathogen, from this plate and place it in the mouse. When we do that, if this mouse then proceeds to develop the disease and ultimately dies, then we have even more confidence that this bacteria that we're studying, the one that we think is a pathogen, we have more confidence that it really is a pathogen. And we have one more postulate and no more room, but I'm going to squeeze it in here. And this is that the pathogen from this dead mouse has to be re-isolated and recultured. So from this dead mouse, we're going to take some of that blood sample again, and we're going to look at it, and we're going to look at it under a microscope. And we're going to see some red blood cells, but we're also going to see the pathogen. We're then going to take this sample and we're going to put it on a petri dish, just like we did down here, And if we do that and allow enough time for growth to occur, and we see this bacterial cell forming these colonies, then we can be confident that this bacteria that we've been studying, that we were calling a presumed pathogen, is indeed a pathogen. And this has been a classic way that we test various agents to see if they cause disease. And it's still the same way we do it for over 100 years. Cook's postulates laid the groundwork for what we call the germ theory. And so let's talk about the germ theory next. So let's begin with a working definition for the germ theory of disease. This theory states that infectious diseases are caused by specific microbes in the body. These microbes can be bacteria, viruses, fungi, prions, all those things that we talked about before. Being an infectious disease, that means that the disease has to be spread from one individual to another. And we can think of disease spreading in one of three different ways. The first is that it can be passed from person to person. It can be passed from person to person by any personal contact. Now, that could be anywhere from standing close enough to someone where particles from one person can move to the other, and it could include sexually transmittable diseases. COVID is transmitted this way from person to person. Some infectious diseases are transmitted by bites from animals. Some of the most deadly infectious diseases are caused by bites from insects, particularly mosquitoes. The third way is through ingesting contaminated foods and water. And it could also be from other environmental sources. Maybe it's in the soil or something like that. By our understanding of Cook's 4 postulates and the acceptance of the germ theory of disease has driven much of modern day medicine. The number of people dying from infectious diseases is much less today than it was 100 years ago, or even 50 years ago. So now I want to talk about why some bacteria are pathogens and other bacteria are not. There are even some species of bacteria that are pathogenic, and other variants, types of that species, are not pathogenic. And I want to talk about 2 adaptations that bacteria have developed that leads them to becoming pathogenic. The first I'm going to talk about are toxins. And then I want to talk about endospores. So let's first talk about toxins. And a good example of how toxins can make a bacteria pathogenic is to look at E. coli cells. Now, E. coli cells can live in certain environments, even in our human body, and are not harmful. In fact, they are quite helpful in our bodies. So in general, E. coli does not cause us problems. However, there are some strains of E. coli, such as the variant that we call 0157H7, and these are harmful to humans. When you hear about an E. coli breakout in the news, it's a good chance they're talking about this variant here. So why is this variant of E. coli, this H7 variant, so much more harmful to us than the one we usually see in our bodies? In today's modern technology, it's easy to sequence these 2 types of E. coli and see if there are any differences. And what was discovered was that this H7 variant has a gene for a toxin. A toxin is nothing more than just a poison that a microorganism produces. And usually toxins in small doses can have a really big impact. This gene is found in this H7 variant, but it is not found in other types of E. coli that do not cause harm. So what does this toxin do? Let's draw a human cell here. And it's usually going to be an intestinal cell, because usually you catch the H7 pathogen from eating contaminated food or contaminated water. So this is going to be an intestinal cell. And then let's draw the E. coli variant here, H7. It will enter the cell, and once it's in the cell, it will produce that toxin. Because remember, there's a gene for the toxin in this E. coli cell, so it'll then make that protein which serves as a toxin. I'm just going to draw the toxin like this. How does that toxin eventually kill this cell? It kills it by going after the ribosomes. So here's a ribosome in this human intestinal cell. And remember, ribosomes, their job in eukaryotic cells, which include humans, in bacterial cells, in archaeal cells, doesn't matter which kind of cell, ribosomes are responsible for making proteins in the cell and ultimately allowing the cell to live. So this toxin, what it does is it binds to the ribosome and blocks it so the ribosome can no longer function. So by producing this toxin, prevents any further protein production in these intestinal cells, and that will lead to cell death. Your intestinal cells are responsible for lots of things, but one thing they do is they help regulate water transfer between our intestines and our body. When these cells die, you get a lot of water that's released in the wrong direction. And it, of course, kills all these cells. And this will lead to bloody diarrhea and can ultimately lead to death. Often people come in contact with this form of E. coli due to poor sanitation, or you eat food that was not properly cleaned, and then you ingest this E. coli cell. So when you hear on the news that there is an E. coli breakout of a particular kind of food or restaurant, please listen to that advice and don't eat that food for a period of time until the FDA tells you it is safe to eat. Okay, next let's talk about endospores. And I have a bit of a confession to make. I recorded this whole presentation here and realized I did not hit record on my camera. So instead of redrawing it, I'm just going to leave it here and I'm going to walk you through it. And as I'm walking you through it, I hope that gives you enough time to draw everything down as well. So a good definition of an endospore is a tough and thick-walled dormant structure. In this case, it'll be a structure from a bacterial cell. And these form in times of stress. So for instance, if a particular bacterial species finds itself in a place with few nutrients, it might form one of these endospores so it can survive. And as I say at the end here, these endospores can survive for thousands of years. They're just waiting for that opportunity to germinate and grow again. So how do these form? Well, over here we see our bacterial species here with its DNA in it, and it's going to divide. And you're going to have two copies of the DNA. Now what normally happens is that this is going to get pinched off under the control of binary fission and produce two new cells that will now divide again and again and again. However, when there's these times of stress, what happens is one of these copies of the genome will form a membrane around it, and also it contains a peptidoglycan layer like you would expect in a bacterial species. We call this the forespore. The other copy of DNA doesn't do anything. Then what happens is a much thicker spore coat forms around the forespore. And this thicker and more protective coat that forms is then released from this bacterial species into the environment. It's important to remember that it's not just this one cell in this colony of millions or billions of cells. There are several of these bacterial cells that are doing this at the same time, so that many endospores are going to be produced and released into the environment and wait, maybe for 1,000 years. But maybe they'll only wait for a week, right? They're just waiting until times are better. Now, this in itself isn't necessarily a bad thing because it does allow a bacteria to survive to fight another day. However, it can be a bad thing for humans or other animals when these bacteria can lead to infection. And I'm going to just tell you two examples. And one is the bacterium that produces the toxin anthrax. So if you breathe in one of these endospores that contains the bacteria that produces anthrax, or if it lands on an open wound, then these endospores will begin to produce that bacterium that can make anthrax, and that can lead to sickness and, in some cases, death of the organism that contracted it. Another example is the bacteria that can lead to gangrene. Gangrene is when you have an infection in a wound, usually a wound, and it doesn't heal because the bacteria begin to grow in it, preventing blood flow to that wound and preventing necessary immune response to protect and heal that individual. This bacterium also forms an endospore, and when that endospore lands in an open wound, that's when you can begin to develop the condition of gangrene. And that can lead to limbs having to be amputated, and it can also lead to death. Now I want to talk a little bit about antibiotic resistance and biofilms. Antibiotics were first discovered in 1928. But it wasn't until the 1940s and to present day, through present day I should say, when they were used widespread. In fact, it was probably World War II that really upped the production of antibiotics. They needed to make sure there was enough antibiotics that were on hand to fight infections before a land invasion in France could occur. They knew that they would take more casualties during the war due to infection than they would from gunfire. So making sure they had enough antibiotics on hand was critical in the war planning stages of World War II, particularly during the D-Day invasion. From that moment on, they began to be used widespread. And in fact, it led to a bit of a problem, and still leads to a problem, because often they are overprescribed. And when they become overprescribed and overproduced, I should say, this can lead to antibiotic resistance. In fact, there's this one very interesting and a little scary study that was done that found bacterial populations in certain soils that used antibiotics as a nutrient. So antibiotics certainly weren't killing these bacteria. It's the reverse. They were taking that antibiotic and they were using it as food. So this has become a very big problem in the medical field, in the agricultural field, lots of fields that had previously used lots of antibiotics to treat patients or to treat livestock and the like. I now want to talk about biofilms. And first, define them, it also tells you how they are also linked with antibiotic resistance. So biofilms are a dense population of bacteria surrounded by polysaccharides, sugars. And this protects them from antibiotics. Having this very thick polysaccharide surrounding and protecting this population of bacteria prevents the antibody from being able to reach the bacteria and killing the bacteria. One specific problem is that we will see these biofilms growing on medical supplies. Such as catheters. And so if you can't treat and get rid of the bacteria on these catheters, that can lead to an increase in urinary tract infections. We'll see biofilms all over the place, and sometimes they cause a problem, sometimes they don't. But like your cell phone likely has some biofilms on it. All right, next I want to talk about something we call bioremediation. And bioremediation is the use of naturally occurring or engineered microorganisms to either consume or break down pollutants. So essentially what this means is we identify an area that is heavily polluted, and we find bacteria that can consume or break down that pollutant, and it cleans up that area. And so I'm going to talk about a few examples here. The first is when we use it to clean up contaminated soil. Most of this contamination, if not all of the contamination that we find in soils, comes from human use. Sometimes it's certain industries that contaminate the land around their plants. Sometimes there's a toxic spill and it contaminates the soil. But we have bacteria that when we put it on the soil will consume this contaminated soil. You may remember back in 2010 when the Deepwater Horizon oil rig failed. So this was an oil pump in the Gulf of Mexico, and when it failed, it released huge amounts of oil into the Gulf Stream. And this ended up decimating that area, killing a lot of wildlife and natural habitats for those animals and plants. We have bacteria that when you put them in an oil spill, they will consume that oil and they will break it down. It doesn't happen overnight, it does take some time, but it is a very natural way to clean up these oil spills. We use bioremediation in crime scene cleanup. So after a crime has occurred and detectives have done their investigation, that material left behind, be it blood or be it semen or something else, has to be cleaned up. Now it's important to clean this up well because that blood or semen may contain various pathogens, either bloodborne pathogens or pathogens that are found in the semen or what other body fluid might be left behind. One could clean up these areas using chemicals. However, we've learned that certain bacteria, when you culture them and place them in these cleanup areas, they will decontaminate it and not put any humans at risk in cleaning it up. These are just 3 examples of bioremediation. There are many other examples. I encourage you to read up on this. Some of them are really interesting, but that's all we have time for right now. All right, that's all I have for today's material. If you have any questions at all, please make sure you reach out to me and I'll do my best to answer any questions you might have. If not, I will see you in class. Bye for now.